In this morning lecture, I want us to focus on specifically one of these excitable tissues, and that is the nervous tissue. We'll then talk about propulsion tissue perhaps next week on Friday. So today we're just focusing on nervous tissue only. I thought I wouldn't want to combine the two so that I don't rush you through the process. And I just checked on our course outline and I realized that we do have enough time in physiology, so we can just spread them out, not a big deal. The specific things we are going to learn in this lecture are the following. In as much as we talked about tissues being consisting of cells, matrix, and tissue fluid, when we talk of nervous tissue, there's nothing much to say about the matrix, the extracellular matrix that is, and the tissue fluid. So we'll only focus on the cellular components of the nervous tissue. And we are going to see how these cellular components are classified. One of those classifications are the neuroglial cells. For this, we are going to state their components as well as indicating the functions of each of those. For neurons, we will focus on their structure and their functional types. And perhaps that's what the anatomy part of the lecture entail. I am more interested in the physiology part of the lecture, where we are going to then describe the cell membrane electrical potentials as they exist in neurons. You remember we talked about excitable tissues. We talked about cell membrane potentials some time back. And we described 3 types of membrane potentials. Whatever we discussed was relatively general. The good thing is that the model we took was largely for neurons. So in simple terms, we're just going to revisit that story of electrical potentials in the cell membrane, but now narrowing it down to specifically for neurons. We are then going to look at how neurons transmit signals from one neuron to another. Now that would be new. We haven't done that one. And it's a very fundamental part of physiology or of medicine in general. And we're going to see why. There'll be 2 types of junctions, what we call electrical junction and what we call chemical junctions, or others called synapse. So we'll see the differences between chemical and electrical synapses. That will be it for today. So let's just start straight ahead by looking at the components of nervous tissue, and specifically we are looking at the cellular components of the nervous tissue. When you look at the cells of the nervous tissue, we can group them into 2. We have the neurons and the neuroglial cells. These are the 2 cellular components of nervous tissue, neurons and neuroglial cells. Neurons are considered the functional cells of the nervous tissue. On the other hand, the neuroglial cells are considered the supporting cells of the nervous tissue. So what this statement means is that neurons are the ones which transmit the signals, because that's the primary function of the nervous system. They're the primary cells that transmit signals within the nervous system. On the other hand, neuroglial cells just provide support to these neurons This support could be in different ways, as we are going to see shortly. Apart from their role, if you want to distinguish these two cell types, the other thing you can distinguish is their excitability. Now, we defined excitability as the ability of a cell to respond by generating an action potential. The ability of a cell to respond to a stimulus by generating an electrical impulse. So on this note, the neurons are the only ones that can do that. The neuroglial cells cannot generate electrical impulses. In as much as we are saying they are part of the nervous tissue, they cannot generate electrical impulses. The other thing we compare is the ability of these cells to renew themselves, the capacity for mitosis. So on this note, we can say that neurons are largely non-renewing. And I put that with a pinch of salt because it doesn't apply to all neurons in the body, but it applies to most neurons in the body. Almost all, because it's over 90% actually. Most neurons do not divide. As a matter of fact, the neurons you're born with could be more than the ones you have right now, because immediately you are born, there are those that actually died. So neurons are largely non-renewing, to mean they are in the G0 phase of the cell cycle. On the other hand, neuroglial cells are mitotic. They do divide. That means they do regenerate themselves. Tailored to that also is based on the numbers of these cells. Neurons are fewer than neuroglial cells. At the time you're born, maybe for every single neuron, you have about 10 neuroglial cells. So even from the beginning, the neuroglial cells are more. But we've said something. Despite the fact that when you're born, the neuroglial cells are more than neurons, we are also saying that the neurons are actually dying, reducing in number, while the neuroglial cells are actually increasing in number because they are mitotic. So for this reason, we will have then more neuroglial cells. At birth, for every one neuron, you have about 10 neuroglial cells. And in adulthood, for every one neuron, you have about 50 neuroglial cells. The concept here is that neurons are fewer than neuroglial cells. The neuroglial cells outnumber the neurons. These are the major differences between neurons and neuroglial cells. I want us to then talk about the neuroglial cells first. We classify neuroglial cells based on where they are found. They can either be within the central nervous system or within the peripheral nervous system. The ones within the central nervous system are therefore simply called the central neuroglial cells. It means they're found within the central nervous system. And if we talk of central nervous system, what do we really mean? That's a question to you. I have disabled the voice thing because there was some noise in the background. You can type. what is central nervous system? I've asked you a question, or you can't type. Okay, maybe one of you can type so that I know that you're able to type, even if you can't type the correct answer. Okay, at least you can type. Okay, good. So Camila, that's the answer. When you talk of central nervous system, we mean the brain and the spinal cord. The brain and the spinal cord. That is central nervous system. So we are saying central neuroglial cells are found within the brain. They are also found within the spinal cord. There are 4 types of central neuroglial cells. We have astrocytes. Astrocytes are the most abundant of the central neuroglial cells. This is an example of an astrocyte. What are their key functions? They provide structural support to neurons. Having said that, they are actually the most abundant. They provide structural support to neurons. Apart from that, now, this structural support is usually in adults as well as even during development, actually. When neurons migrate, they anchor themselves on astrocytes. But even in adulthood, wherever they are, they're anchored by astrocytes. Astrocytes also form part of what we call the blood-brain barrier. You realize that neurons are a bit sensitive, or let me say very sensitive. And so anything within the blood should not necessarily reach the neurons, otherwise it will affect neurotransmission. It might even generate abnormal signals within neurons. For that reason, the body has a way of controlling the contents within the blood that can actually reach neurons. That control is achieved by what we call the blood-brain barrier. The blood-brain barrier is an anatomical barrier. It is also a physiological barrier, and it's actually also a biochemical barrier. It's a barrier that basically obscure some substances within the blood from reaching neurons. Astrocytes contribute part of this barrier. Also, when there is injury in the central nervous system, neurons, we've said, that cannot really regenerate for healing. So what happens is that astrocytes are the ones which can undergo mitosis and contribute to healing or repair. And that's what I'm calling healing by gliosis. Basically, it means repair. Last but not least, astrocytes modulate neurotransmission. Neurons communicate with one another through what we call synapses. in as much as astrocytes are not excitable cells, astrocytes can modify the biochemical contents within a synapse, therefore influencing how neurons communicate with each other. And that's what I mean by modulating neurotransmission. These are not the only functions of astrocytes, but I really consider them key. There are other functions that you can check in the anatomy books, physiology books, and they're still okay even if you write an exam. There are many functions of astrocytes, but at least remember these four. They're easy to remember as well. The second type of central neuroglial cell is what you're calling the oligodendrocytes. Oligodendrocytes look like this, okay, or maybe in this image, that one is what is represented as oligodendrocytes. What is unique about them, or what is their role? Oligodendrocytes form the myelin sheath around axons within the central nervous system. That is what we call myelination. This is myelin sheath. That's myelin sheath. That's myelin sheath. That's myelin sheath. So this wrapping around axons of neurons is what we call myelination. And oligodendrocytes are the one which perform this role as long as you are within the central nervous system. Perhaps what is important to take note of is that a single oligodendrocyte actually myelinates multiple axons of neurons. So this single cell is wrapping around this axon. It's also wrapping around this axon. That is not to mean that it's the only one that's wrapping around that axon anyway. We can see that a single axon is also being myelinated by multiple oligodendrocytes. But the point I want to highlight here is that a single cell can wrap around multiple axons within the central nervous system. That will be a bit different from what you're going to see for the peripheral nervous system. The third central neuroglial cell is what we call the ependymal cells. Ependymal cells line the brain ventricles. Apart from the brain ventricles, they also line the central canal of the spinal cord. Now, the brain ventricles are cavities which contain fluid, the cerebrospinal fluid. The central canal does the same, but it's in the spinal cord. So these cavities contain cerebrospinal fluid. The ependymal cells line this cavity, and usually ependymal cells actually secrete that particular fluid, the cerebrospinal fluid. Apart from secreting the fluid, the ependymal cells also promote the flow of the fluid. This is because ependymal cells have cilia. So the ciliary activity of the ependymal cells can promote the flow of cerebrospinal fluid. The last central neuroglial cell to talk about is the microglial cells. As the name suggests, they are a bit smaller compared to the others. This is microglial in this particular diagrammatic representation of these cells. So the microglial cells are a bit smaller than the others. That's why we call them microglial cell. Microglial cells are unique cells of the CNS. basically they are macrophages within the central nervous system. And just like any other macrophages in the body, they therefore come from bone marrow. They are cells of bone marrow origin, except they'll invade the CNS and be there. So they are macrophages within the central nervous system. I remember giving assignments on naming macrophages on one column and where these macrophages are found on the other column as you match the two columns. So one of the things that should be in your columns is microglial cell on your left hand side and central nervous system on your right hand side. I believe you've done the other cells. You've already listed that. So anyway, because microglial cells are macrophages within the central nervous system, they therefore offer defense against infective organisms. In simple terms, they have a role in preventing against infections. This role is immunity. They're the immune cells of the central nervous system. Apart from that, because of their phagocytic role, microglial cells can also, excuse me, because of their phagocytic role, the microglial cells can also clear debris within the central nervous system. So these are the central neuroglial cells. Now let's flip the coin and talk about the peripheral neuroglial cells, which means neuroglial cells within the peripheral nervous system. So here we are going to talk about two types of cells. The first one to talk about are the Schwann cells. Schwann cells are like oligodendrocytes in terms of function, that now they form myelin sheath within the peripheral nervous system. Remember, oligodendrocytes form the sheath within the central nervous system. These ones form myelin sheath within the peripheral nervous system. What is important to note, even from this image, is that a single Schwann cell can only wrap around a single axon. So a single Schwann cell can only myelinate one axon. Again, that is not to say that one axon is only myelinated by a single Schwann cell. As we can see here, there are multiple Schwann cells myelinating this axon. The gaps between the wraps are known as the nodes of Ranvier. Towards the end, okay, maybe in the middle of this lecture, we are going to talk about the nodes of Ranvier, and we are going to understand their importance. Maybe before I go to that, so I have perhaps highlighted on the differences between myelination within the central nervous system and myelination within the peripheral nervous system. And I want some 3 points to be clear to you. One of the differences between central and peripheral myelination is that in central myelination, we're using oligodendrocytes. In peripheral myelination, we are using Schwann cells. Another difference is that in central myelination, oligodendrocytes can wrap around multiple axons. In peripheral myelination, Schwann cell can only wrap around a single axon. The third difference in terms of central and peripheral myelination is more about the type of proteins within the myelin sheath. Let me just keep it simple and say that the type of proteins that constitute the myelin sheath, well, the myelin sheath is actually lipid largely, but there's some proteins within this lipid. They're lipoproteins. The proteins within the myelin are different for central and peripheral myelination. The proteins are different. I'll just leave it to that instead of giving you the names of the proteins. But basically, because of the difference in the types of proteins, we will see some diseases that only affect the central nervous system. We'll see some autoimmune diseases that only affect the peripheral nervous system. Yet they both attack myelin sheath. So those three differences, one is based on the type of cells involved. The second one is based on how these cells myelinate. And the third one is based on the type of proteins. I am avoiding to give you the names of the proteins, so just grasp the concept. Right, the second cell type that we consider as peripheral neuroglial is what we call the neurosatellite cell. This neurosatellite cell can simply be called the satellite cells. Satellite cells surround cell bodies of neurons within the peripheral nervous system. Like here, these are cell body of a neuron within the peripheral nervous system. The satellite cells surround their cell bodies. We believe that by being in that specific location, the neurosatellite cells provide structural and nutritional support to the cell bodies of neurons within the peripheral nervous system. They provide structural and nutritional support to cell bodies of neurons outside the central nervous system. If you are outside the central nervous system, you are in the peripheral nervous system. So you can compare satellite cells or neurosatellite cells to astrocytes, except that now astrocytes are in the CNS. These ones are in the PNS. Great. We've talked about neuroglial cells. We've seen 6 types of neuroglial cells. There are other cells which can still be considered as neuroglial in quotes, peripheral especially, but they're not very classical, and so I've chosen to avoid them. But in your reading, you may come across multiple other names considered as peripheral neuroglia, apart from Schwann cells and neurosatellite cells. Even if you do, there's no problem, but if I ever ask you to name peripheral neuroglial cells, you must mention these two first. Then I don't care what else you want to mention. Now let's talk about neurons. Neurons have some 2 key functional properties that I want to explain. One of them is that neurons are excitable. Neurons are excitable cells. What does that mean? It means that they can respond to impulses they can respond to stimuli by generating bioelectric impulses. And these bioelectric impulses are the ones you're calling action potentials. So excitability is a property of neurons. The second property of neurons is conductivity. Conductivity is the ability to transmit electrical impulse, usually along the cell membrane of that particular cell. So neurons can generate impulses. Neurons can also transmit these impulses along the cell membrane. So far, the basic tissues we've done, the other two, none of them could do this too. And so neurons are very unique. We are going to see maybe next week that the other cell that can actually do these two are muscle cells. Right. I want to give you some 2 minutes to answer this question. Whatever you do, I'm giving you 2 minutes. Whether you're just staring at the screen or whether you actually draw in your book. I don't even know whether you have a book. These online things are very interesting. Or wherever you just Google and see how it looks like, it's okay. So whatever you do, just do it. 2 minutes. So I believe you've already drawn. Whatever you've drawn, I'll ask it of you one day. Let's proceed now. Now, from what you've drawn, there are some things that need to come out. I know you've drawn something different from what I'm going to show you, and that's the point. Here, I'm just going to show you the very basic structure, but I know you've done something better than this one. What I'm going to draw, what I've drawn here is largely focusing on these functional domains of a neuron. So from your diagram, these three must come out, plus others that I'm not going to mention. There are three functional domains of a neuron. One of these domains is what we call the receptive domain of a neuron. You didn't call it receptive domain, and that's okay. I didn't expect you to call it receptive domain. As a matter of fact, it's not called receptive domain. I am just approaching this from a different perspective so that I don't repeat what you've already done. The receptive domain of a neuron refers to these ones, which we call the dendrites. So in your diagram, you must have illustrated the dendrites. The receptive domain of a neuron refers to the dendrites. The role of the dendrites is to receive signals either from the environment or from other neurons. Usually the dendrites have multiple branches so that you can increase the receptive field, the surface area over which impulses are detected. The second functional domain of a neuron is what we call the integration domain. The integration domain of a neuron refers to this one. This is what we call the cell body of a neuron. So you must have called it the cell body of a neuron, or you must have called it the soma, S-O-M-A, soma. In plural, somata. The cell body of a neuron is the one that contains those organelles, and therefore we call it the integration domain because the one that basically internalizes the functions of a neuron. Now, one of the key functions of the cell body is to produce or to synthesize the neurotransmitters. The neurotransmitters are synthesized. If they are proteins, they'll be synthesized through that process of protein synthesis, the one you did assignment on, through that process of protein synthesis. And usually they will then be transported along the axon to the end of the neuron. But first, they are synthesized within here, the cell body. Cell bodies can have different names depending on where they are found. A group of cell bodies within the peripheral nervous system, if you have several cell bodies grouped together within the peripheral nervous system, you call that a ganglion. A ganglion is a group of neuronal cell bodies outside the central nervous system, which means within the peripheral nervous system. If you are to have a similar concept now within the central nervous system, then that's what we call the gray matter. So gray matter is a collection of neuronal cell bodies inside the central nervous system. And that gray matter can either qualify to be called cortex or a nucleus, depending on where it is located within the central nervous system. If it is in the periphery, the outer parts of the central nervous system. For example, it's in the outer layer of the brain, then you call it cortex. But if it's deep inside in the brain, you call it nucleus. For now, just remember that when you have several cell bodies grouped together within the central nervous system, they constitute the gray matter of the central nervous system, which can either be considered as cortex or nucleus. Don't confuse nucleus of collections of cell bodies of neurons with this nucleus here. Same term, two different things. The third functional domain of a neuron is a transmission domain, or you can call it the transport domain. This refers to the axon. So we call it transmission domain because the axon is the one that transmits impulses along the cell membrane. We call it transport domain because the axon is the one that transports neurotransmitters from the cell body to the end of the neuron. When you have, maybe it's important to note that the size, the diameter of the axon varies. There's some neurons with thin diameter of the axon, and there's some neurons with a wider diameter of the axons. The wider the diameter of a neuron, of the axon of a neuron is, the faster it can transmit impulses. So based on the size, based on the diameter of the axons of neurons, we have different types of neurons. There are those that we'll call alpha neurons, beta neurons, delta neurons, gamma neurons. So they are based on the size. Now, a simple way of classifying them based on the diameter of the axon is to just call them type A, type B, and type C neurons. A being the widest, B intermediate, C are the thinnest of them all. All in all, when you have axons grouped together, they constitute the white matter of the brain. So when you talk of white matter, you're referring to axons of neurons. When you talk of gray matter, you're referring to cell bodies of neurons. How do we classify neurons? We classify neurons based on different concepts, but I'll teach you two. One of them is based on the structure of the neuron. So we call this the structural classification of neurons. Structurally, we classify neurons according to the number of processes that arise from the cell body of that neuron. For example, here, we see a neuron here with 5 dendritic processes and 1 axonal process. So we can say this neuron has 6 processes arising from its cell body. If the number of processes arising from the cell body of a neuron are more than 2, then we call such a neuron a multipolar neuron. In this second example, we see a cell body of a neuron here with 2 processes arising from it, one process being an axonal process and the other process being a dendritic process. So this neuron has 2 processes arising from it. We therefore call it a bipolar neuron. Compare those two with this one. In this one, we have a cell body of a neuron with a single process arising from it, so we may want to call it unipolar neuron. However, look at what happened to that unipole. The unipole then divides into two. So this neuron is a player. A single one, but again divides into two. One of the process is dendritic, the other process is axonal. This neuron is therefore called a pseudo-unipolar neuron. It's a false single pole. The fourth type of neuron based on this classification is what you'll call a polar neuron. A polar neuron, one word, a polar neuron. A polar neuron will be just a cell body without any process arising from it. Maybe we start with that one. Apolar neurons are neuroblasts. Neuroblasts are cells which are going to become neurons. So we see them in embryonic tissue. We'll be talking about embryology more from next week. They are embryonic cells which are going to become neurons. So before these neurons form their processes, they'll just be rounded cells. We call them apolar neurons. How about pseudonipolar neurons? We have said that pseudonipolar neurons, where the cell body is here, there's a short single process which then divides into two. Usually these two, there'll be one process which is peripheral, and that's the one that is dendritic. And there'll be another process that is central. That's the one that will be axonal. Pseudounipolar neurons are found in the peripheral nervous system. These neurons form the primary neuron that transmit sensations from receptors of general sensations. So they are primary sensory neurons from receptors of general sensations. When you talk of general sensations, it's different from talking about common senses. That older concept of 5 common senses, you'll realize it's really null and void when you come to medicine. There are more than 5 senses and they're all common. Either way, so when you talk of general sensations, what do we mean? We mean things like pain sensation, temperature sensation, touch sensation, pressure sensation, vibration sensation. Those 5 are examples of general sensations: pain, temperature, touch, pressure, vibration. Those are examples of general sensations. The neurons that carry signals from receptors of such sensations look like this. They are pseudounipolar neurons. So it means that the dendritic process will be the one on the receptor side, peripheral, and then the axonal process, the one that will be going to the spinal cord or to the brain, the central process. That means that their cell bodies will not be within the central nervous system. The cell bodies will be outside the central nervous system. Their cell bodies are the ones which constitute ganglia. Ganglion in singular, ganglia in plural. We've also talked about bipolar neurons, that they have 2 processes, one being dendritic and the other one being axonal. Bipolar neurons are also primary sensory neurons, but they'll be primary sensory neurons from receptors of special sensations, such as retina, which is special sense receptor for vision. Vision is special sensation. The cells within the olfactory epithelium are also bipolar. Olfactory epithelium is the receptor for smell. Smell is also a spatial sensation. You will learn later that sensations can be classified as general or spatial. Spatial senses are 5 only. General sensations are more than 5. Spatial senses include vision, smell, taste, hearing, and equilibrium of the head. Those are spatial sensations. General sensations are more than that list. And that's what I was telling you. This concept of 5 common senses is null and void here. So bipolar neurons are found in the retina, spatial sense for vision. They're also found in the olfactory epithelium, the spatial receptor for smell. Multipolar neurons is where there are several dendrites arising from the cell body, in addition to the axon also arising from the same location. We find multipolar neurons as interneurons. Interneurons are neurons which join 2 or more neurons within the central nervous system. So multipolar neurons are interneurons. Also, multipolar neurons are motor neurons. Motor neurons are neurons which supply effectors, neurons that arise from the central nervous system and go to effectors. Effectors being either muscles or glands. So neurons which come all the way from the central nervous system transmit signals from the CNS to effectors which are either muscles or glands are considered motor neurons. These motor neurons are multipolar. So this is the structural classification of neurons. How about the functional classification of neurons? Functionally, we classify neurons based on the direction of the nerve impulses. Now, this direction is with respect to the central nervous system. If the neuron is carrying impulses towards the central nervous system, like this neuron is doing, then we'll give it the name an afferent neuron. An afferent neuron takes signals from the periphery to the central nervous system. These signals are therefore likely to be sensory, and for that reason, afferent neurons are sensory neurons. On the other hand, now you realize that we've just used a pseudonipolar neuron for that purpose, because this receptor, or rather this organ is the skin here, so most likely it's carrying information regarding general sensation like pain or touch, so it's a pseudonipolar neuron there anatomically, but functionally we call it afferent neuron. Now on the other hand, this one here is carrying impulses from the central nervous system to the peripheral, to an effector, in this case muscle, and structurally you realize that that is a multipolar neuron, but functionally we call this neuron efferent neuron. Efferent neurons will be going to either muscles or glands, so efferent neurons are motor neurons. The third example would be a neuron that joins 2 or more neurons within the central nervous system. It doesn't have to be part of a reflex arc like this one. As long as you have 2 neurons within the CNS, could be even this one and another one here. As long as you have 2 neurons being connected by a neuron, you call such a neuron an interneuron. An interneuron is also known as association neuron, a neuron that joins two or more neurons within the central nervous system. That is how we classify neurons functionally. I told you that the other way in which you can classify neurons is based on the diameter of the axons, based on the axonal diameter. So based on this, we can have type A, type B, and type C cells. The things I mentioned about alpha, beta, delta, gamma are actually subtypes of type A neurons. I don't want you to go into the details of how big is a type A and how small is a type C. The only thing I want you to remember is that this variation is based on the diameter. So the wider ones will be A and the thinnest one will be C. And that means that the A ones are the ones which will transmit signals faster. Good. I want to give you a break. So maybe let me check my watch. So we are now at 9:08. I can give you 10 minutes break. So we come back at 9. So let's come back at 9:20. Take a break. We come back at 9:20. From there, we are going to talk about the membrane potentials in neurons. It will require that you're a bit fresh, so I'm giving you a small break. Let me unmute you so that if you have a question, you're also free to ask. So I believe we should be back, or at least we should continue. We are now going to look at largely physiological aspects of nervous tissue and specifically neurons. We'll start with the membrane potentials as displayed by neurons. So I hope you remember that there are three types of membrane potentials: the resting membrane potential, graded potentials, and action potentials. The resting membrane potentials for neurons is at about -70 millivolts. I hope you remember the one for cardiac muscle is -85, and for skeletal muscle -90. You also remember that we have graded potentials. Graded potentials are short-lived. They are focal disturbance of the membrane. But if they reach the threshold, they can initiate action potentials. The threshold for initiating action potentials in neurons is 15 millivolts plus minus 5. So it will vary. So maybe what we can say with confidence here is that the thresholds which are less than 10 millivolts will not generate action potentials. So if the membrane potential is at minus 70, if you have a disturbance that makes it to, let's say, 61, that will not generate an action potential. If you have a disturbance that at least disturbs it for about 20 millivolts, that will definitely generate action potentials. So if it's at negative 70, if it's at negative 70, if the disturbance makes it go to negative 50, it will definitely generate an action potential. Now I want to ask a question to you. If the resting membrane potential is at negative 70 and you have a graded potential that disturbs the membrane by about 20 millivolts, so making the membrane potential to move from negative 70 millivolts to negative 90 millivolts, you've moved with a difference of 20 millivolts. That particular graded potential will not generate an action potential. My question to you is why? You can unmute yourself, then you answer. Why wouldn't a graded potential that makes the cell membrane potential move from negative 70 millivolts to negative 90 millivolts, why wouldn't such graded potential initiate an action potential? I believe one day the answer will come. Action potentials of neurons last for about 2 to 4 milliseconds. Well, we've said that the threshold... Yes, please.
Because there is it is hyperpolarization.
Very good. Thank you, Kevin. We said that action potentials can only be generated by depolarizations. A hyperpolarization cannot generate an action potential. Now, action potentials in neurons last for about 2 to 4 milliseconds. The time span between the origin of an action potential and its end is about 2 to 4 milliseconds. Very short-lived. Well, we'll see that in muscles even shorter. For skeletal muscles, it's about 1 to 2 milliseconds. For cardiac muscles, very long, about 300 milliseconds, and we'll see why. So here we're just talking about the neurons. The speed at which neurons will transmit impulses will vary depending on whether the neuron is myelinated or not, and the size of that neuron in terms of the axonal diameter. So the larger neurons are the ones which transmit faster. If the axonal diameter is wide, it will transmit signals faster. If the neuron is myelinated, it will also transmit signals faster. So myelinated neurons with wider diameter will be the fastest. Unmyelinated neurons with narrower diameter will be the slowest. The range at which signal is transmitted along the axon fiber varies a lot. Could be as low as 5 meters per second, could be as fast as 120 meters per second. So the range is wide. All right, so it's that time that I want to take roll call. You'll be discussing this question as I take the roll call. So I'll divide you into your groups. I am the one creating the group, so don't worry, just accept the group I'll have given you. Okay. So join your groups and do that question. I believe you people are back now. Some of you are very unique. I start taking the roll call when I have 50 people. By the time I'm finishing, I have 65. It's okay. I'll take another one later. At a time I'll not warn. So I hope you've discussed that we have, sorry. So I hope you've discussed that we have multiple phases of neuronal action potential. This is the resting membrane potential. Then you have the depolarization phase. This is what we called the overshoot. This is the repolarization phase. And this is the hyperpolarization phase. In terms of molecular explanations for them, you remember that for the resting membrane potential, there were 4 factors to consider that explain the resting membrane potential. Potassium efflux is several times faster than sodium influx. Then we have sodium-potassium pump. You have anions, and lastly, the fact that chloride influx is also there. The depolarization phase is explained by opening of the voltage-gated sodium channels so that we now have sodium influx. The overshoot is due to closure of the voltage-gated sodium channels. The repolarization phase is due to opening of voltage-gated potassium channels so that now we have potassium efflux. Then lastly, the hyperpolarization phase is due to delayed closure of the voltage-gated potassium channels. The fact that these channels close at around -80. So when the membrane potential reaches -70, the channels are still open, and that is why it will continue beyond -70. We'll then expect to see this one only in neurons. We don't expect it in muscle because of their level of resting membrane potential. Now, action potentials in neurons are propagated along the axonal membrane. The cell membrane wrapping around the axon is the one that propagates the action potentials. And these action potentials are the ones responsible for transmitting nerve signals. So signals are transmitted along the cell membrane of the axon. What is the effect of actually myelinating an axon? Now, myelinated neurons have nodes of Ranvier. Consider this to be a myelin sheath, and this is another myelin sheath. Doesn't matter whether it is within the central nervous system or in the peripheral nervous system. We'll have a node of Ranvier there. Magnified will have this as a node of Ranvier, this region here. So this is myelin sheath covering an axon. This is another myelin sheath covering an axon. But this region where we have node of Ranvier, there is no myelin sheath there. That's the point. Regions of nodes of Ranvier lack myelin covering. That means that regions where we have nodes of Ranvier are exposed to the extracellular fluid or the interstitial fluid. But regions covered by myelin sheath are not exposed to the extracellular fluid. Now, remember what happens in depolarization of neurons. During depolarization of the cell membrane to create an action potential, voltage-gated channels are the ones which open, then now sodium rushes in. If this region is not exposed to the extracellular fluid, then it means even if the voltage-gated sodium channels open here, you will not have a sodium rush at this point. But here, we'll have sodium rush at that point because there is sodium here. And so we'll have a sodium influx at that point. So what does that mean? It means that in myelinated neurons, sodium influx can only occur at the nodes of Ranvier. Sodium influx cannot occur at the myelinated region. But at the nodes of Ranvier, sodium influx will occur there during depolarization. Now, the implication of that statement is that when an action potential is therefore experienced by the neuron, the action potential will then be jumping, in quotes, from one node of Ranvier to the next, because the depolarizations, or rather the sodium influxes, are just happening at the levels of the nodes of Ranvier. It means that now action potential or depolarization events will be jumping from one node of Ranvier to the next. This jumping of action potential from one node of Ranvier to the next is termed as saltatory conduction. So myelinated neurons display saltatory conduction. The importance of saltatory conduction is twofold. One is that it will increase the velocity of signal transmission. The velocity at which action potential is moving along the cell membrane is increased if the action potential is jumping from one node of Ranvier to the next. But the second advantage is that you realize that if the action potential is just jumping from here to here, so this region here, the membrane is actually not affected much. For that reason, there'll be less energy required to return this membrane to normal, this region. And so we say that because the depolarization events are just at the nodes of Ranvier, then this segment of the membrane, because it is less affected, there'll be less energy required to return it to normal. Saltatory conduction conserves nerve energy. So this is the twofold advantages of myelinating neurons: increased speed of conduction and also conservation of neuronal energy. These are the advantages of saltatory conduction. Now, what happens? We have said that action potentials are transmitted along the cell membrane of the axon. What happens when that signal reaches the end of the axon? And now we want to transmit this signal from one neuron to another. In simple terms, we have a junction. What happens at the junction? At the junction between one neuron and another, we must have a way in which the signal will be transmitted. And that's what we want us to look now. Now, junctions between one neuron and another are termed synapses. A synapse is a junction between one neuron and another. The term synapse simply means connection. There are two types of synapses in neurons. There's what we call the electrical synapse. What is an electrical synapse? An electrical synapse is where the two neurons are joined by a gap junction. So if you join two neurons by a gap junction, what will happen? Look, this is a neuron. The depolarization is coming like this up to there. Then there are gap junctions joining this neuron and the next. What will that mean? If we have depolarization here, it means that we'll have sodium influx here. So when sodium influx here, massive, that sodium can easily diffuse through the gap junction to the next cell, which we're now calling the postsynaptic cell. If sodium influx is here that has caused depolarization, the sodium here will diffuse easily through the gap junction to the cytoplasm of the next cell, because these are just gaps. You know, gap junctions are channels where the two cytoplasm concerned are actually in free communication. So ions can diffuse freely from one to another. If sodium diffuses to this one, it means that this neuron will then also initiate an action potential of its own, because it will have a disturbance of the cell membrane potential. If that reaches threshold, this neuron will initiate an action potential, and that is how the signal will have been transmitted from here to there. So electrical synapses are very fast, because it just requires ions to move from one cell, the presynaptic cell, to the other cell, the postsynaptic cell, through those gap junctions. If the ions move, then the depolarization will spread from one cell to another. And once you initiate depolarization in the postsynaptic neuron, you are good to go. So that neuron will again carry the impulse until the end. And so that is how electrical synapses work. How about the second type of junction transmission, which I'm calling chemical synapse? Now, there are things you need to understand about a chemical synapse. So look at this. This is an axon that has reached here, then synapse with this neuron, maybe via multiple points. So we start another neuron here. That is a neuron. Again, that neuron reaches and synapses. So this is the axon, but this is the dendrite of the next neuron. Axon synapses now with the dendrite. Remember, the dendrites are the receptive domain. The axons are the transport domain. So we want to see what happens here. What happens here? So we say that this one can be a gap junction, which is okay. That means that when you have depolarization here, the ions here can diffuse freely through gap junctions to the next one. And that is what we call electrical synapse. How about in a chemical synapse? In a chemical synapse, there's some terms we need to define first before we talk about that chemical synapse. In a chemical synapse, we have what we are calling the presynaptic cell and a postsynaptic cell, or now that they're neurons. So with reference to this synapse, this cell is the presynaptic neuron, and this is the postsynaptic neuron. But with regard to this is the presynaptic, and this is the postsynaptic. I hope you understand what pre and postsynaptic neuron mean. There's also another term to define. This is the synaptic terminal. The synaptic terminal is the end of the axon. So for this one, this is the synaptic terminal. For this one, this is the synaptic terminal. The ends of the axon of a neuron are the synaptic terminals, or what's called axonal tips. The other term to define is pre- and postsynaptic membrane. In the understanding that this axon, of course, is a cell, so it has a cell membrane. So the cell membrane of the axonal terminal, or the cell membrane of the synaptic terminal, is what we are calling the presynaptic membrane. The cell membrane of the dendrite that is receiving at the synapse is what we are calling the postsynaptic membrane. Maybe at a higher magnification, this is the presynaptic membrane, this membrane here. And this is the postsynaptic membrane. There's also what we call the synaptic cleft. The cleft is the space, the extracellular space between adjacent pre- and postsynaptic membranes. The space. Now remember that space is almost equal to the interstitial space. The cleft between the two adjacent membranes is what we're calling the synaptic cleft. Then there is what we call the neurotransmitter. The neurotransmitter is a chemical that facilitates signal transmission from one cell to another. The chemical that facilitates signal transmission from one cell to another is what we're calling the neurotransmitter. Now, if you're using the term neurotransmitter, then it means that the cells producing that chemical must be neurons, and the ones receiving it also must be neurons. You remember we talked about types of ligands, talked about paracrine ligands, talked about hormones, neurohormones, neurotransmitters. So neurotransmitters where the synthetic and the secretory and the receptor cell are both neurons. Lastly, there's what we call the synaptic vesicles. The synaptic vesicles are actually secretory vesicles that contain the neurotransmitter. Secretory vesicles that contain neurotransmitter are known as synaptic vesicles. So that means they'll be present in the presynaptic cell, not in the postsynaptic cell. So with all that understanding, let's see how chemical synapse operates. Now let's just follow slowly, but make sure you understand. So we are saying that neurons will experience action potentials. And we are saying this action potential is transmitted along the cell membrane of the axon up to the end of the axon. That end of the axon is what we are calling the synaptic terminal. So let's consider this to be that situation. This is the end of an axon. This is the dendrite of the next cell, the receiving cell. This is the end of an axon. So this is the cell that is sending signal. So first, there will be action potential that comes up to the end of this axon. The action potential, we know how it operates. It is just a depolarization event. It's a depolarization event. Once the depolarization event has reached the end of the axon, what next? That is the question. Now, this is what is next. The depolarization at the end of the axon causes the synaptic vesicles to go fuse with the cell membrane, and that is what we call docking. It will make the synaptic vesicles to dock. This is docking. when they fuse with the cell membrane here. It's called docking. And after they have docked, they release their neurotransmitters. They release the chemicals they contain through the process of exocytosis. So depolarization that reaches here causes that to happen. Now, how it causes that to happen is a bit of a detail that I really didn't want to go into, but it involves calcium ions. Remember, when depolarization comes like this, usually it's largely sodium influx, but at the axonal terminal, we usually have voltage-gated calcium channels as well at the axonal terminal. These voltage-gated calcium channels do open when depolarization reaches here. And when you have calcium influx, calcium is the one that makes these vesicles to move from wherever they are up to the presynaptic membrane in a process we are calling docking. And after they have docked, then you're going to have exocytosis. Just summarize that by saying that depolarization, because whether that depolarization is due to sodium influx or it's due to calcium influx, is still a depolarization event. There are molecular details there which you don't have to go into. But eventually, you are going to have exocytosis of the neurotransmitter. So now, see here, the neurotransmitter which was in the vesicles has now been released into the synaptic cleft. Once the neurotransmitter is in the synaptic cleft, what happens to the neurotransmitter? There are three possible options of things which can happen to the neurotransmitter once the neurotransmitter is in the synaptic cleft. One of those options is that the neurotransmitter, which is in the cleft here, can bind to its receptors. Now, the receptors of the neurotransmitter will then be found in the cell membrane of the postsynaptic cell. The presynaptic cell contains vesicles with the neurotransmitter. The postsynaptic cell contains receptors for that neurotransmitter. So one option that the neurotransmitter will do is to bind to its receptors, which are on the postsynaptic membrane. That is one option. But that's not the only option. The other option is that the neurotransmitter can actually diffuse away from the synaptic cleft. Now that, I'm using the term diffusion loosely here, but basically it means that it can go away from here. It can go away from here because it has been taken up by an astrocyte. That is what we call uptake. It means that the astrocytes take away the neurotransmitter from the cleft. That's what we call uptake. But the neurotransmitter can also decide to diffuse back to the cell that produced it. And that is what we call reuptake. So we have uptake and we have reuptake. Reuptake is when it goes back to the cell that produced it. Uptake is when it goes to another cell, not necessarily the one that produced it. So the neurotransmitter can do that. The third option is that once the neurotransmitter is in the cleft, this neurotransmitter can undergo enzymatic degradation. There are some enzymes that can break down this neurotransmitter. It can be degraded. Those are the three options that the neurotransmitter can undertake. If we are interested in transmitting signals from one neuron to another, then it means that this one and this one is not our interest. Our interest is this one. So let's follow this one, where the neurotransmitter binds to its receptor on the postsynaptic cell. So if you follow this diagram, the chemicals are within the vesicles here. Those chemicals, the vesicles have docked, then the chemicals have been released into the synaptic cleft. Let's consider this to be the chemical. So this chemical, which has been released here, can be uptaken, not important for neurotransmission, can be reuptaken, not important for neurotransmission, can undergo enzymatic degradation, again not important, but also it can bind to its receptors on the postsynaptic neuron. The binding of the neurotransmitter on the postsynaptic neuron is the one that is important for neurotransmission. So what happens? Now, see here, these are the receptors of the neurotransmitter. The receptors of neurotransmitters in the postsynaptic membrane are actually ligand-gated sodium channels. Now, not voltage-gated sodium channels, but ligand-gated sodium channels. The receptors of neurotransmitters on the postsynaptic membrane are ligand-gated sodium channels. What does that mean? It means that the channel is usually closed, but can open if the right chemical binds on it. The right chemical here is a ligand. For voltage-gated channels, the channels are usually closed, but if there is electrical disturbance that is significant enough, it can cause the cell membrane, it can cause the channel to open. Those are voltage-gated channels. For ligand-gated channels, they're always closed, but if there is a chemical messenger that binds to that particular receptor, or rather to that particular ligand, that particular channel, then the channel can open. So here, yes, these proteins are receptors, but also they are channels. Which type of channels? Ligand-gated channels. When we talked about cell membrane proteins, we said that a protein can have multiple functions. And this is a good example. It's a receptor for a particular ligand. That ligand can be any neurotransmitter. It could be adrenaline, for example, or noradrenaline, or acetylcholine, whichever it is. So once the ligand has bound to its receptor, it will make these receptors to open because they are ligand-gated channels. If the ligand-gated channels open, what will happen? What will happen is now sodium will enter into the cell. So I've just noticed, by the way, that the green ones are actually sodium molecules, and I don't know which color is this. For lack of a better term, allow me to call them brown. The brown ones are the neurotransmitters because they're here. So the neurotransmitter binds into its receptor, and the binding of the neurotransmitter into the receptor causes this receptor to open, because this receptor is also a ligand-gated channel. So the receptor opens its channel, and once it opens its channels, sodium ions will now enter. That is sodium influx. If you have sodium influx, what will be the outcome of that? So these ligand-gated sodium channels operate the same way as voltage-gated sodium channels, except that their triggers are different. That means they'll open, and that means you're going to have massive sodium influx, which the potassium efflux cannot overcome, and therefore, in simple terms, you are going to generate an action potential. It will be a depolarization that causes an action potential in the postsynaptic membrane. Now, that was the main aim. We wanted a way in which action potential here can move to the other one, and now we finally have an action potential in the postsynaptic neuron. That is how signals are transmitted in neurons. We've gone through electrical synapse. We've also gone through a chemical synapse. I'm not so sure whether you've understood the chemical synapse, but I want to repeat it again briefly in a different way so that you understand. Because chemical synapse is an important part of medicine. You cannot afford not to understand chemical synapse in neurons if you're going to understand anything pharmacology. So, in a nutshell, we've said that for signals to move from this cell to this cell, first of all, an action potential will have to arrive at the end of the axon. Once the action potential has arrived at the end of the axon, then it will cause voltage-gated calcium channels to open at the end of the axon. And that means you're going to have calcium influx. When you have calcium influx, the synaptic vesicles are going to move to the cell membrane in what we call docking. Once the vesicles have docked, then they will release their neurotransmitters, their contents, into the synaptic cleft through the process of exocytosis. The neurotransmitters within the cleft can be degraded, can go back to the original cell, or can go back to another cell. But all those are not important for transmission. The one that is important for transmission is if the neurotransmitter then binds to its receptor on the postsynaptic cell. The receptors of the neurotransmitter are on the postsynaptic cell. This is the receptor for the neurotransmitter. Once the neurotransmitter is bound to its receptor on the postsynaptic cell, these receptors, which are also ligand-gated sodium channels, will then open. If they open, then we are going to have massive sodium influx in the postsynaptic cell, and that will mean that we'll have depolarization in the postsynaptic cell that will cause generation of another action potential in this neuron. So a signal has been transmitted. Now I want to give you a question as I take the next attendance. So the question is this. I can see I'm starting 57 and I'll keep it locked. Whoever comes in will not join. Outline the differences between chemical synapse and electrical synapse. That's a question. Discuss that in your groups. The groups are open. All right, you should be back now. So I want us to finish this lecture by looking at something. Before that, take that assignment. You'll do this on your own, at your own time. So we'll just list 10 examples of neurotransmitters. I'll not ask you to submit it to me. Just do. I'll ask you one day. I think in this class, I've given you a few examples. I mentioned adrenaline. I mentioned noradrenaline. And I mentioned acetylcholine. So you can add 7 others. All right. What's the importance of junctional transmission in neurons? The importance of junction transmission in neurons can be applied in pharmacology, in the manufacture of drugs, in the giving of drugs, and this is what you'll be doing, and that's why it's very important that you understand this. Now, in terms of pharmacology, you remember this is chemicals, so what we are interested in here is more of the chemical synapse, the importance of chemical synapse in medicine. When you want to manufacture drugs, there are 2 things to think about. You can have a drug that promotes the effects of the neurotransmitter, but you can also have drugs which reduce the effects of the neurotransmitter. Let's take an example of a chemical synapse that uses adrenaline as the neurotransmitter. So in that previous chemical synapse we've described, suppose the neurotransmitter there is adrenaline, and that is part of the normal physiology of that particular region. We can manufacture drugs that influence that transmission. So you can have drugs which will promote the effects of the neurotransmitter, or let me say mimic the effects of the neurotransmitter. You can use the term mimic instead of promote. But you can also have drugs that cancel the effects of the neurotransmitter. So I want to give you examples of such drugs. Then you'll be telling me, will those drugs mimic the effects of the neurotransmitter, of which they'll now be called agonistic drugs? Agonistic drugs are drugs which mimic the effects of the neurotransmitter. Or will those mechanisms be mechanisms that cancel the effects of the neurotransmitter? And we use the term antagonistic drugs to mean that. Is that drug agonistic or antagonistic? Okay, for the sake of discussion, we can just choose to use our mic instead of the chat so that I am able to follow. I'm usually a bit slow in these things, so I may not be able to coordinate the chats and the talk. So we'll go with, if you have a response, just unmute yourself and then give an answer. Now I'll give you options. One of the options is we can have a drug that obscure the binding site of the neurotransmitter on the receptor. Tell me, if we have such a drug, will that drug have agonistic effect, which means mimic the effects of the neurotransmitter? Or will such a drug have antagonistic effect, which means it will cancel the effects of the neurotransmitter? Anyone?
Let me try.
Okay, Anthony, carry on.
Yeah, the drug will have antagonistic effect.
Okay, you mean this one, eh?
Yeah.
Okay, very good. And that's true. So if you have such a drug, it will have antagonistic effect, which means it will block the effects of the neurotransmitter. Another option. If you have a drug that prevents docking of the synaptic vesicles, and there are drugs which do prevent docking of the synaptic vesicles, what will these drugs do? Will they have agonistic effect, which means this, or will they have antagonistic effects, which means this? Anyone?
I can try.
Okay.
It will have a antagonistic
Very good.
effect.
That's very true. So such a drug will also have antagonistic effects. It will block the effects of the neurotransmitter. Another example, we agreed that once in the synaptic cleft, there are some 3 options that can happen to the neurotransmitter. It can bind to its receptor, it can be degraded by the enzyme, and it can also be taken by the cell that produced it, that is called reuptake, or taken by another cell. So if you have a drug that inhibits the degrading enzyme, what will that drug do? Yes, Ashley?
It's an agonistic.
Very
drug.
good. That drug will promote the effect, so it will be agonistic drug. Another example, if you have a drug that binds to the receptors of the neurotransmitter, what will that drug do? Think about that first. What will that drug do? Anyone?
Trial.
Okay.
Antagonistic.
Okay, I agree with you, Kwanzu, but it's optional. It can be both. It depends on what that drug actually does. There are drugs which bind to the receptors and then block the receptor. Those ones will be antagonistic. But there are those drugs which bind to the receptors and stimulate the receptors. Those ones will be agonistic. So when you have a drug binding to a receptor, you want to still ask yourself, what does the drug do on that receptor when it binds? So the binding itself is not the main thing here. What is important here is, what does that drug do on that receptor? It can bind and block. So you'll be calling such drugs receptor blockers. But there are some drugs that bind to the receptors and stimulate them. So when a drug binds to the receptor, it can either have that effect or that effect. Good. 2 more examples. If you have a drug that inhibits the reuptake of the neurotransmitter. What will that drug do? Anyone?
Let me try.
Yes, Devix.
It will antagonistic.
It will be antagonistic. That may not be true. The reason is because if you inhibit reuptake, it means that the neurotransmitter will be more available within the synaptic cleft. And that means that more neurotransmitter will be available to bind to its receptors on the postsynaptic membrane. So therefore, a drug that inhibits reuptake will be agonistic. One of the best types of antidepressants are actually doing this one. They inhibit reuptake of serotonin. So you have more serotonin stimulating you to make you happier. They are antidepressants. Serotonin is the one that makes you jumpy. Okay, one last one. If you have a drug that promotes exocytosis of the synaptic vesicles, what will...
Let me try.
Yes, Camilla, try.
Agonistic effect.
Sorry, it will have?
An agonistic effect.
Very good. It will have agonistic effect. And the reason is because, again, more neurotransmitters will be available for signal transmission. Good. That is the application of chemical synapse in pharmacology. I am now done with the lecture that I wanted to give today. If you have any question, you can ask now.